This video describes the procedure developed by the HIV Vaccine Trials Network at Fred Hutchinson Cancer Research Center for staining fresh whole blood in multicenter clinical trials to be analyzed by a centralized laboratory. The procedure utilizes TrueCount tubes made by Becton Dickinson Biosciences, each of which contains a lyophilized pellet that dissolves during sample preparation to release a known number of fluorescent beads. Fluorescent antibodies are used to stain for surface markers that are specific to different cell populations. The number of cells in these different populations can be compared to the number of fluorescent beads during flow cytometry analysis, and absolute concentrations of different leukocyte populations in whole blood can be tracked over time in relation to intervention.
Hello, my name is Erica Andersen-Nissen, and I'm a staff scientist in the HIV Vaccine Trials Network. The procedure that you're about to watch is one that we've developed for use in our multi-center clinical trials. In these trials, blood processing laboratory technicians receive fresh whole blood specimens, stain them with fluorescently labeled antibodies, and then ship the specimens to a centralized laboratory that's capable of analyzing them on the flow cytometer. Fresh whole blood is often not analyzed in the context of clinical trial settings due to cost and feasibility issues, but we've developed a single antibody staining panel that can identify the major leukocyte populations in the peripheral blood. And staining for these populations takes under 45 minutes, making it feasible for use in clinical trials. In the HVTN, we're using this panel to understand the early events that occur after vaccination with candidate HIV vaccines, but this panel could be further modified to focus in on cell